Hi, morning.
Good evening, everybody.
ערב טוב.
Good evening again. Let's wait for another minute. We'll start. So as usual, let's start with asking, how are you doing this evening? Great. In pain, I'm sorry. I hope this is not because there is no government as of yet. All right, let's move on.
Can you show the the number again for
The number?
Yeah, I missed it.
So again, which number are you referring to?
For joining the main time.
Oh, well, there will be another questionnaire later on.
Oh, okay.
We'll see.
Okay.
But, yeah. Let's move on. All right, so let me remind you about the assignment. I got lots of questions via email, so I want to go over this again. So we want you to pick up a paper, and it's not just a paper. It should not be a paper from your own research area that you know and are familiar with, and it's easy for you to work on. We want a story, an interesting story. Again, this is not a regular class where, you know, everybody present their own work or their own fields. I want you to get a little bit out of your comfort zone and find us a very good and captivating story. I'm going to give you an amazing story next time. And hopefully you can find as good stories as yourselves. So yeah, it can be a very good paper. We went through very good 2 papers in the class, like Watson and Crick and Forster. But you really have to come up with a good reason why you want to cover a very good paper in a field, fundamental paper that started the field. And I think I already authorized one such paper. But it will make much a better story if you can find a worse paper in your field or in other fields. A famous fraudulent science case, famous retraction, errata. Basically, you need to captivate the rest of the class with a very good story, with basically a lesson that everybody can take home. We want this course to really teach us how to write good papers and avoid bad science. And there are so many cases of bad science, and I think we can all learn from those stories. So this is basically the assignment. We set up a Google Doc, and let me share that for a second.
Sorry, there are 2 students that are trying to get in to the class.
Okay. For some reason, I didn't get an alert. Everybody can see the Google Sheet?
Yes.
Okay. So we are going to have, at this point, there are 31 students registered for the class. I don't know if anybody will quit, but basically we have 7 dates, and we are planning for 4 people to speak in each of these dates. That's 28. If you are going to accommodate 31, we will need 3 of those 7 dates to have 5 presenters rather than 4. And most importantly, we need people to occupy, the meeting in December 6 and December 13. Obviously, you're coming first, so we'll take this into consideration. But obviously, people who have not registered as of yet don't have much choice, and they will have to populate these dates. So I encourage you to hurry up and reserve a date and put in a link for the paper that I authorize. So you all need to communicate with me about ideas of what you're going to present, and I'm going to authorize. Question about this? So again, please fill in the Google Sheet. I need you to commit for a date. I need volunteers to populate the early dates, especially December 6, December 13. And we will probably need to accommodate up to 5 speakers for some of these dates. Regarding another assignment, you're going to get today an assignment, which will be basically questions about the paper I'm going to present in class today. And I would like you to submit your work by midnight on Sunday, this coming Sunday. So you basically need to answer a few questions. This is paper outside of your field. And really the exercise is to learn how to read a paper outside of the field, trying to, put your best effort to try to understand the paper. And I'm sure it's going to be quite difficult for many of you because you are not familiar with the topic at all. So try your best. You're not going to get graded on wrong answers. You're just going to get graded off putting in the effort. So I don't expect you guys to become biophysicists and really answer everything correctly, but I do expect you to put in the effort, and that's what you're going to be judged on. Any questions about this?
Yes, so we need to read an article that you will upload and then answer some questions?
Correct.
Okay, thank you.
And there might be a problem for our meeting on November 20, I think it's 29 actually, not 28, not 29. I have to check the calendar. Are we meeting on the 28th or on the 29th? Which one is it, Tuesday?
29, it's 14 days from
okay,
today.
so that's the correct date. The reason is I'm actually en route coming to Israel. I'm arriving on the 29 at 4 p.m. and our class is at 7. I asked Khaled to talk to Rita. Did you talk to her already? No. So in principle, we should get a room. Last quarter, we did this in the big room next to the department's office on the 3rd floor. I hope we can reserve this room for 7 p.m. for basically 29, December 6, and December 13. Those are the dates that I'll be in Israel, and we will have a hybrid format for those 3 classes. However, if for whatever reason my flight is delayed, we'll have to cancel this class on the 29th of November. I'll be in touch with Khaled, and Khaled propagate the information. I hope to make it. I will basically rent a car and drive directly from the airport to campus. And I hope that, you know, this plan will work. If it doesn't work, we'll have to find, you know, to pack more people on the remaining classes. Any other questions about administrative stuff? Okay. So what we covered last time was a Forrester paper and a little bit analysis of that paper by Bob Clegg, and basically the basic theory and the basic equation that comes out of this theory by Forrester. And what we are going to do today, that was really serving as a segue for my own research, and I'm going to cover single molecule FRET that my group developed and has become very popular by many, many groups. And basically, what I want to show you today is the power of this approach. So we already discussed that fluorescence resonant energy transfer is a radiation-less transfer. In contrast, for example, to electron transfer, Markov theory, here we are talking about dipole-dipole interactions. There are no electrons moving around. And just the fact that 2 dyes, one is excited, the other one is not, are near each other, the excitation can actually magically hop from one to another, but it obeys this famous Forrester formula of distance. So the rate of energy transfer is 1 over the lifetime, excited state lifetime of the donor, times this parameter R0 called the Forrester radius divided by the distance to the 6th power. So again, we excite the donor transfer energy to acceptor, and we collect photons from the acceptor. Actually, we collect photons both from the donor and the acceptor. And the ratio of how many green photons and how many red photons we have really tell us about the distance between those 2 molecules. So it's a ruler. We can measure distances on the nanometer scale with this spectroscopic ruler. So a key paper in the field. So Forrester theory is 1946, 1948. There are quite a bit of spectroscopy studies done in the 50s and the mix up to mid-60s. But really the paper that changed the whole landscape of the story of FRET is a paper by Lubert Stryer and Dick Hageland, Robert Steyer was a very famous biochemistry professor at Stanford University. Dick Hageland was a PhD student. Later on, he went to start a company called Molecular Probes, which was later on bought by Invitrogen and then Sigma and LifeTech. And nowadays it's basically Sigma, and this is how you buy fluorescence dyes, indicators, and so on. This is the company of Dick Hageland. In the 2000s, he sold his company, bought a nice yacht, and went all over the world. They published in 1967, in May 29, just a few days before the Six-Day War, this amazing paper. It was communicated by Art Kornberg, a Nobel laureate, and his son is also a Nobel laureate, and he is part-time at Hebrew U and in Stanford. And this paper really demonstrates that FRET can be used as a molecular ruler. We report here an experimental study of the dependence of energy transfer on distance. The observed efficiency of energy transfer as function of distance is an excellent agreement with R to the 1 over R to the 6th power, as predicted by Forster. And what they've done, they basically synthesized peptides, polyproline peptides. So it's many repeats of proline. And the repeats went from 1 to 12. So they had a monomer of proline, a dimer, trimer, and all the way to 12 units conjugated together as a single peptide. And they had this electron donor, a dye as a donor and a dye as an acceptor on the two ends of the different lengths peptides. They measured the energy transfer for all 12 samples and then basically studied the dependence of energy transfer on distance. And this is the results. So they plot here basically the transfer efficiency. We'll talk about how you measure transfer efficiency as function of distance. And distance is basically translated to the number of monomers conjugated to the peptide. And they have a fit here. And if you plot it on a log-log scale, you find that the slope is 5.9, really, really close to 6, the predicted value by Forster theory. So basically, they demonstrate that at least in this range of distance around, let's say, 20 to 40 angstroms or 2 to 4 nanometers, there is almost a linear behavior between distance and energy transfer. And therefore, energy transfer can be used as a ruler to measure distances in this range. The reason you have this almost linear behavior here is that 1 over R6 is a very steep function, and you have this range where you have almost linear behavior. So, well, that was really a paper that totally changed the field, because until then it was really just theory, and maybe people who did spectroscopy kind of paid some attention, but suddenly because of this paper, the whole biochemistry community started to put dyes on proteins and on nucleic acid and started to measure distances. That was a big shift, and from that point on, FRET becomes a really important tool in biochemistry. Mind you, it's all at this point done on the ensemble level. So you prepare a sample, you purify your proteins, you label your proteins, you put it in a cuvette, you go to the fluorometer, and you do measurements. So this is how it was done. And again, the function here, this is just a caption that discusses the fitting function, and then they get 5.9, again, confirming the Forster theory. I was totally unaware of, and most of the community is unaware of, that actually somebody did work before them. This is Mayor Walczyk from Weizmann. And in fact, there was this competing group, eventually, this is really the group of Ephraim Katzir, who was the first president of Israel, who continued to work on Forster and did some fantastic work on FRET in competition with the Stanford group. They did not get as much credit as the Stanford group. And what is really amazing, what you see here is a very similar experiment to Starr and Hagoland that was done a few months before, but it's not showing the full curve, it's just showing one tail, one side of the 1/R6 dependence. And this is what they say here in their paper. Quenching was observed in the series of tryptophan glycine 0 to 4 monomers with ionization of the phenolic group. The efficiency of quenching decreases with increasing molecular size. Quenching is attributed to radiation, less energy transfer between the tryptophan and ionized tyrosine residues. So basically, they look at the same phenomena. And this is published in February 67. Mind you, the paper of Starr and Hagoland was published in May. And that is basically 3 months before. And obviously, Stryer was very famous, was connected properly, published in the right journal. Many, many reasons. The Stryer and Hagoland paper is much, much more well-known to the community compared to this paper. Questions so far? Okay. So the minute Fred becomes a molecular ruler, demonstrated as a molecular ruler, it becomes very, very important. And many people use it. It allows to make measurements both in vitro and in vivo, measure distances. It has near angstrom resolution, very sensitive. You basically excite donor, collect photons from donor acceptor, energy transfer efficiency goes by this formula. And practically what that means, if you are able to do a spectroscopic measurement and look at the spectrum of the pair, let's say your protein is labeled with donor at one position, acceptor with another. You have many proteins in your cuvette, you put it in the fluorometer, and you measure spectrum, and the combined spectrum should show 2 peaks. One peak coming from the donor and one peak coming from the acceptor. And depending on the weight of those 2 peaks, you can actually extract the distance. In the case where the distance between donor and acceptor is very far, then you'll see mostly emission from the donor because there is very little energy transfer and very little emission from the acceptor. In the case that are very close, the donor emission is almost totally quenched, and you see mostly acceptor emission. And at R0, the distance, which is called Forster radius, where you have 50% energy transfer, that means R equals R0. So this here basically is 1/2. You get basically equal contribution for donor and acceptor. So that's a ruler. You basically do a spectroscopic measurement, and you get distances. And this could be used for many, many different things. First of all, just measure distance, as you've seen on polypeptides. You can actually look at molecular interactions. If 2 components come and bind, they are free in solution, and you can actually even measure binding constant by following this measurement as function of time. So you start at a large distance, and eventually after they bind, you get a short distance. You can look at enzymes that cut proteins or cut nucleic acids. You start with a Fred pair, and you cut, and then you see the individual components. You go from short distance to large distance. And you can do this with fluorescent proteins, by fusion. You can do many different things. The next development in the field is our contribution, which is going from ensemble level to single molecule level. So what do you gain by going to the single molecule Fred measurement? Let me give you a simulation here. So let me unshare for a second. Everybody can see the simulator?
Yes.
Great. So this is actually a simulator that my colleague Xavier Michelet, who has been with me for almost 25 years, wrote about 25 years ago when we just started. And I still really like to use this simulator. What you see here is a 2-dimensional test tube where we throw into the test tube, let's say, linear DNA or linear peptide, where each of the ends of those linear oligomers is labeled with donor and acceptor. We excite with a laser the donor, and we collect photons from donor and acceptor on the ensemble level. So basically on the two detectors, you'll see here green and red signals over time. You see that they have fixed value, and I can use the first formula to calculate the distance at each point in time, and basically I get one value, and that value is 1/2. Because we basically set up the parameters to be at Foster radius. However, if I'm starting to reduce my molecules in my test tube, instead of having 128 molecules, now I have 8. You now start to see that the fluorescent signal from donor and acceptor is fluctuating. And instead of having one value for FRET efficiency, which was 1/2, now I get a distribution. And if I go to the limit case of one molecule in my test tube, I can actually look at fluctuations that correspond to distant changes on that one molecule. When I'm on the ensemble level, I cannot see those fluctuations. They are totally hidden. So FRET was used until we demonstrated the feasibility to do this on single molecules. FRET was used to look at averages, at average distance over population and over time. But the minute you go to the single molecule level, you can now look at moving parts of molecular machines. And that's basically the message that I'm trying to convey here. Going to single molecule FRET allows you to look at moving parts of macromolecules. So let's stop sharing this. Second. Okay, so how to measure single molecule FRET rather than ensemble FRET? In the early days of single molecule biophysics, people believed that you cannot really use a conventional microscope with points per function, diffraction limited points per function to excite a single molecule and collect enough signal, because the background signal, the autofluorescence, scales with the volume of the excitation. A signal from a single molecule is more or less constant depending on the light excitation intensity. If you have a very large volume of excitation, the signal will be fixed, but the background will be very high because of autofluorescence. And if you reduce excitation volume to smaller and smaller volumes, you keep the signals fixed, but you reduce the autofluorescence. And therefore, the idea was to use very small excitation volume to look at single molecules. The first demonstration of doing this at the single molecule level at room temperature was Eric Betzig. Later on, he got the Nobel Prize for super resolution imaging in 2014. And before him, W.E. Morner, who shared the same prize in 2014 for single molecule detection at cryogenic temperatures. And the thinking at that time was really that one needs to limit the volume. So a good way of limiting the volume of excitation was using a very small aperture. If you illuminate a very small aperture in the near field, where immediately after that small aperture, you can get a very small excitation volume. Now, a way to implement this idea of having a small aperture in a metallic screen was actually not doing this as shown here, but actually doing it with optical fibers, using heat to pull and break the fiber in a way that you get a conical shaped tip, coat that fiber with aluminum and just the apex being exposed, forcing light through the fiber and getting a little bit of light coming out here. You have to bring this very close to your sample. That requires some kind of a feedback loop that allows you to monitor the distance between the end of the tip and the sample. And this is basically called the near field microscope. The idea was really developed by Aaron Lewis, who later, he was in Cornell, and Eric Betzig was his PhD student. Aaron made Aliyah and came to Hebrew U. I actually got to know him during my PhD just before I left to Bell Labs. And Eric finished his PhD and moved from Cornell to AT&T Bell Labs and continued to develop this concept. He developed the fiber as a way to implement the aperture. He developed a feedback loop called shear force feedback to keep the tip very close to the sample. And in 1993, he published a key paper, which is actually cited in his Nobel Prize. This is a figures taken from this science 1993 science paper, where you can see here a field of single dye molecules. And since the aperture is implemented through a fiber, the electrical field lines coming out of this aperture, if you look from the side view, look like this. If you look from top, looks like this. And since dye molecules have a dipole moment, as you excite and scan the dipole moment of the dye molecule, you can actually get different shapes that really describe the PSF of this aperture. And you can get many cases. If you have a flat dipole on the surface, you get this type of PSF. If it's vertical to the surface, you get two crescents, and so on and so forth. So what Eric was able to show in this paper is the orientation of all these molecules in the field of view. But the fact that he was able to measure single molecule at room temperature was really a big breakthrough, because nobody believed that could be done. Turned out eventually that you actually don't need the near-field microscope. You can use a conventional lens, and you work in the far field using confocal microscopy or total internal reflection microscopy. And it's much easier than what it was thought to be. But at that time, that was a huge breakthrough. And since we built a near-field microscope at that time for other purposes, I was studying excitonic phenomena in quantum wells, and we wanted to look at excitons. We just finished building our microscope, and Eric published his paper. So the first thing we did immediately was to throw a few dye molecules on the surface of a glass cover slip and put it under the microscope, and sure enough, we could see single molecules. So this is data from my lab. It's not even published. What you can see here, we are basically raster scanning the near-field tip from top left to bottom right, like this. And you see single molecules, and they show up pixel by pixel. If you zoom in, you can see here a nice stable molecule. That's A. If you look at B, you see a molecule that basically photo-bleached halfway through. Here you see a molecule that had a dark state. And here you see a molecule that is very noisy. All of these phenomena later on were used for super resolution imaging. We need blinking, we need turning on and off and so on. And that was already established in those very early days, mid-90s, once people started to look at single molecules. Okay, so we asked ourselves, what can we do that Eric has not done? And the idea was to bring two molecules one next to each other. And this is what you see here. Instead of what Straw and Hagelin did, which was peptides, what we did was to use DNA. That was done with the help of Paul Selvin, now a professor in Urbana-Champaign. And we basically attached a donor molecule, tetramethylrhodamine, TMR, to one end of the DNA, and another molecule called Texas Red on the other end. Here you see the spectrum from the pair. In solid black, you see the donor excitation spectrum. In dashed orange, you see the emission. And in solid magenta, you see the absorption of the acceptor. And in red, dashed red, you see the emission of the acceptor. And the hashed green area here is basically something called the spectral overlap. We'll talk about this in a few slides. And as I mentioned, we used the near-field microscope to do this first measurement. Later on, we basically abandoned the near-field microscope and just used a conventional objective in a conventional confocal microscope. By the way, you can stop me at any point. If anybody has a question, please turn on your mic and ask a question. Everything is clear so far? All right, so in 1996, we published a paper where we demonstrated, indeed, we can measure threat on the single molecule level on this DNA construct that I just showed you. That was published in PNAS. And here is the key result of that paper. We basically use a spectrometer. We coupled the light from the near-field probe. We collected all the fluorescent light from the probe, went through a grating and CCD camera on the spectrometer and collected spectrum from a single pair as function of time. So time starts here at 0. And you can see the spectrum from the pair for 5 minutes. You see a drastic event at 3.5 minutes, the spectrum that looks mostly like acceptor spectrum switches and becomes donor spectrum. And at 5 minutes, the donor bleaches as well, and what you're left with is just autofluorescent background. So if we now, instead of doing this detection with a full spectrometer, okay, so what I've just shown here, I'm focusing on the 3.5 minute, 3.5 minutes time point. You see a frame in red. This is mostly the acceptor spectrum with a small shoulder on the donor. And then there is another frame, the next frame, which is kind of intermediate. Something happened in this intermediate frame. And then the following frame just shows the donor spectrum. So something happened here in 3.5 minutes. This is basically the bleaching of the acceptor. We could see also cases on other molecules on the surface where actually the donor bleaches first, and then you're left just with acceptor direct excitation. And if you, instead of using a spectrometer, you just use 2 point detectors, avalanche photodiodes, and you raster scan the sample, then you see basically you can overlay the detection from those 2 detectors, false color them in green and red, and you can see some yellow spots. Basically, you see photons both from donor acceptor, and all of them basically start red and switch green. It's the same phenomena that you have seen before, now shown on single module, on single detectors. This is the detection of the red photons, of the acceptor photons on this APD, shown here in red, and this is the detection of the green photons. And you see when the acceptor photo bleaches, the donor emission goes up. So I would like you now to answer the following question, which is why acceptor photo bleaching gives a proof for single molecule threat. And you can now see the code for the Mentimeter here.
We still see the presentation.
You don't see the Mentimeter?
No.
No.
Sorry. Okay. Can you see this now? Okay.
Yes.
Can you see the presentation now? Or still just a Mentimeter?
Just the Mentimeter.
And now?
Presentation.
What are you seeing now?
The presentation.
Okay, and so I'm trying, and do you see Mentimeter now or presentation?
The presentation still.
I'm trying to share both, that doesn't work that well. Let's just do Mentimeter for now. Just a sec. Can you see this now? Mentimeter?
Yes.
Yes. It shows only one molecule was absorbing all the energy. I'm not sure I understand what you're saying. You can actually open your mic and just give an explanation. My question is why this jump here is a proof for energy transfer.
We see the Mentimeter and not.
Sorry again?
We see the Mentimeter and not the presentation.
Yeah. Well, you know what? Let me try to show them again, both of them at the same time. Okay, now you can see both. Okay, so what I'm trying to understand why this data here is a proof for energy transfer.
Nothing excited the acceptor.
So all the energy goes.
So the energy had to transfer.
Well, what happens when the acceptor photobleaches?
So I'm not 100% sure with what you are referring to with photo bleaching. I'm not from that
Photo bleaching basically
area.
The acceptor molecule degrades. Chemical bonds break. It doesn't absorb light anymore.
Okay, so something, but something made it happen because you didn't excite it with the proper energy, with the proper frequency that matches its band gap.
Yes.
So it had to transfer through the donor because you basically excited the donor, if I'm understanding you correctly.
Right, and then somehow this molecule basically falls apart. So what happens to the excitation of the donor? Basically, when the donor cannot transfer energy to the acceptor anymore, all its excitation has to come out as photons for the donor. So the point is, that when you excite here, you're exciting only the donor. But you see only photons from acceptor. You don't see photons from donor because the donor and acceptor are really close to each other. So you have 100% energy transfer. All the photons go to the acceptor. At this point in time, the acceptor basically blows up. The chemical bonds are destroyed. It's no longer an absorber, but the donor is excited. So what will happen? The donor basically starts to emit because it doesn't have other channels for the excitations. All the photons come through the donor. So that transition here is really a proof that during this time, from 0 to 100, we had 100% energy transfer. And when the acceptor photobleached, all the energy went to the donor. So that's basically a proof for energy transfer. All right. So what I've shown you was the use initially of the near-field microscope on molecules deposited on cover slips. Those are immobile molecules under pretty artificial conditions. And the next level was to do the same in solution. So now we allow molecules to diffuse in a small test tube. So basically we take the microscope, we focus very tightly to a confocal spot inside the solution, We have the two same detectors as before, but instead of creating an image by raster scanning and building a picture pixel by pixel, this confocal spot is now stationary, and molecules are allowed to diffuse in solution, and they come in and out of the confocal spot. And each time a molecules come to the confocal spot, the donor gets excited, transfer energy to acceptor, and then it diffuses out. So what you get on the two detectors is basically time trajectories with bursts. And each burst represents a single molecule event. So now you are actually converting this technology to something very similar to fax machines. Anybody is familiar what fax machine does? Fax is fluorescence activated cell sorting. It's a very highly used technology both for diagnostics and for life science research. You basically take cells from the blood, stain them with dyes, run them through a machine that basically get fluorescent signal from each cell going through the stream. It's basically a fluidic stream with a focused light. And you can build histogram of different cell populations by doing so. In fact, many diagnostic assays, when you go to a regular physical and they draw blood, some of those assays will be done by fax. So what we have done now here, we basically build a fax machines for single molecules instead of for cells. Because if we, within few minutes, we can look at tens of thousands of molecules, assign each molecule a Fred value. So here we prepared several different DNA molecules where the distance between donor and acceptor is different. And you can see, so here you see three histogram, Fred histogram taken in this solution-based measurement. And you can see here when the distance is 7 bases, the Fred efficiency is 1. When it's 12 bases, it's 0.7. When it's 19 bases, it's 0.4. And there is always a peak around 0. This is basically donor-only molecules. They don't have acceptors because when you prepare those samples, there is always some contamination. But the Fred relevant peak is a peak that changes with distance. This is this second peak. And if you do it for enough DNA molecules, you try a very similar experiment that was done by Story and Hagoland on peptides. Now we do it on DNA. So we measure energy transfer efficiencies functions for many different DNAs at different distances. And you can see that the fit 1 over this r to the 6th power is not as good as we have seen for Story and Hagoland paper. This point was actually looked at more carefully by Bill Eaton and Ben Schuler. And what they've done, they really reproduced Story and Hagoland paper on polypeptides, on polyproline. They basically took the exact same molecules. The only thing that is different, Story and Hagoland used very small UV dyes for the donor and acceptor that went on the 2 ends of the peptide. For single molecule experiments, one need to use much better, higher quantum yield dyes. Those are usually larger. They have several different carbon ring configurations. So that's the only difference between Story and Hagoland experiment in '67 and Schuler and Eaton experiment in 2005. And obviously they don't get the 1 over r to the 6th power as well. The observed Fred efficiency agree with those determined from ensemble lifetime measurements, but differ considerably from the value expected from the theory with polyproline treated as a rigid rod. So the assumption was that the polyproline is the best biological molecule that can be used as a ruler because it's rigid and fixed. And they get very different results. So here, again, you see this non-Fred peak, donor-only peak that you can ignore. And here you see the change in the Fred efficiency as function of distance on this different number of monomers attached to the peptide. And instead of 1 over r to the 6th power, they actually get 1 over r to the 4th power. Huge difference. So the question is why? How come the single molecule measurement is so different from the ensemble measurement that was done in 1967? So they were not happy with their first paper, and they went and did another much more careful study in 2007. And they basically looked, what are the reasons for the deviation from the Foster theory? And what they conclude is that 30% of the molecules were basically interconverting, cis-trans isomerization. And also the two dyes are connected to the peptide with soft linkers. We find that fluctuations of the chromophores attached to long flexible linkers also play an important role. So here comes another question. How come Stryer and Haviland got such a perfect fit? They got 5.9 instead of 6. We got something very different on DNA. Bill Eaton and Benchulo got something very different for polyproline. So those single molecule experiments actually are much more precise than what was done in 67. And the question is, how come Stryer and Haviland got such a perfect fit? Did they manipulate the data anyway? We don't know. Nonetheless, the theory is correct. 1 over r to the 6th power is correct. But when you go down to the details and you do a very detailed experiment, you start to find many other phenomena that kind of screw up the goodness of the fitness. But to put all of this aside, let's just say that the idea of now measuring this on small macromolecules and looking at fluctuations allows us to look at moving parts of single molecules. These are really very, very old examples and motivators for what we wanted to do. So, oops, why is it working? Worked before. Huh. I'm sorry. So, okay, something is wrong. We are not going to spend time on this. This was supposed to be a movie of a small peptide folding. And this is a movie of a molecular machine that rotates, and you can see the rotation. And basically the idea is now with this method, you can look at not static structural biology, but at dynamic structure, and basically study moving parts of enzymes, of molecular machines. And indeed, since 96 until nowadays, there are hundreds, if not thousands, of groups doing all kinds of very interesting biochemistry, molecular biology studies, and even in live cells nowadays, using single molecule frames. Oh, here it's now moving. Can you see the rotation? Folding doesn't work. Okay. So what I want to devote the rest of the meeting today is one particular topic of great interest to my own group, which is initiation of transcription. So I think many of you are physicists and engineers, so I don't know if you are familiar with the central dogma of biology. We talked about Watson and Crick and the structure of double helix DNA that obviously immediately explain how cells can replicate their genomic material and cells during cell division. And basically following the discovery of double helix, immediately the central dogma was put forward, which basically states the following: all the genetic material sits on chromosomes in the DNA. When cells divide, you need to make 2 copies of that genomic information. So you have an DNA polymerase that is responsible for this copying. When you need particular proteins, you do it in 2 steps. First, you read a particular gene and you convert it to an intermediate molecule called messenger RNA. The enzyme responsible for this is called RNA polymerase. And my group has been working with RNA polymerase for many years, all the way nowadays to RNA polymerase of coronavirus. And once you have a messenger RNA, the ribosome converts that message into a protein. So that's basically the central dogma. It had many refinements and additions. So one is that proteins don't have an infinite lifetime. They are actually being degraded. They use a whole dedicated machinery to degrade proteins such that you can make them fresh all the time. And many parts of this dogma received pretty amazing Nobel prizes for discovering the structure, understanding mechanisms, and so on. So RNA polymerase is Roger Kornberg. We mentioned his father already. He was the one who communicated striaton paper. And Roger is in Stanford as his father and at Hebrew U also. He got the 2006 Nobel Prize for basically solving the structure of RNA polymerase. You're all familiar with Ada Yonath, who got the Nobel Prize in 2009 for solving the structure of the ribosome. Abraham Hershko and Aaron Jehanover got the 2004 Nobel Prize for the machinery responsible for cleaning proteins. And DNA polymerase also got the Nobel Prize. I just put, Israeli-related prizes here to this central dogma. And let me now describe to you the bacterial RNA polymerase cycle, the transcription cycle for the bacteria. The reason everybody studies bacteria is that that machinery is so well conserved among the whole tree of life. So even the human transcription system, the actual core of the machine is very identical to the machine in E. coli. Of course, for the human system, there are many, many other components. And in addition, the regulation of transcription in humans is much more complicated than in bacteria, but even in bacteria, it's quite complicated. So here you see a bacterial transcription cycle. We start with the protein, the core enzyme called RNA polymerase. RNA polymerase doesn't know how to find a beginning of a gene. The beginning of a gene is called a promoter. Basically, in front of every gene, there is a particular DNA sequence that is recognized by the holoenzyme to come and bind exactly at the right place in order to transcribe the gene. As I told you, the core RNA polymerase doesn't know how to find the start of a gene. It needs another factor. It's called a transcription factor. For housekeeping genes, it's called sigma 70. There are many other sigmas which relate to when you are in stress, when the cells are in stress, you need to express and make proteins of other kinds. So there is, for example, sigma 54, sigma 34, all kinds of different sigmas. But the common one for regular housekeeping genes is called sigma 70. So sigma 70 binds the core enzyme to form the holoenzyme. The holoenzyme is now capable of finding a start of a gene. It binds the DNA, forms something called the closed complex. So some isomerization steps, it opens something called the transcription bubble. It's about 10, 11 bases of a bubble open. So you dehybridize the double-strand DNA into single-strand DNA for about 10, 11 bases. This is called the open complex. And now if you give the machine its fuel, and the fuel for this machine is basically individual nucleotides, it will start to transcribe and make messenger RNA. Actually, in this step, there is something called abortive initiation. For strong promoters, the enzyme doesn't like to leave the promoter. It starts to make transcript, lose them, slide back, try again, try again, try again. And only if there is kind of thermal fluctuation, some energy coming from the bath, from the outside, it overcomes some kind of a barrier, potential barrier, and goes into elongation. And once it's in elongation, it's very possessive, and it will continue all the way to termination, make the full transcript. The transcript is released, the individual proteins fall apart, and all the components are ready for another cycle. My group focuses on this particular part of the cycle, which is initiation. Why that is important? Initiation is the most regulated step in gene expression. You know that everything in our body is orchestrated very well. Proteins are being made as needed for different functions. And there is a whole regulation machinery that is in charge of which proteins are expressed at what time. And all this regulation is happening not at the conversion of messenger RNA to proteins by the ribosome. It's actually regulated at the step of making this intermediate molecule the messenger RNA. So the regulation of gene expression happens here. So this is why we study transcription initiation. We use the method that I described before, which is single molecule, usually in the format of molecules diffusing in solution. So what I've shown you before here is a molecule that diffuses through the confocal spot. We excite the donor only with one laser, donor transfer energy to acceptor, and we collect photons both at the donor and the acceptor wavelengths. We calculate for each burst a Fred value. Here you see burst on the two detectors, and that translates into histograms I've shown you before, one-dimensional histogram. So here you see the DNA with short distance. This is actually a measurement where we make a mixture of those two different DNAs. You see two peaks, and you see the zero peak, which comes from contamination, which we want to get rid of. It's always there, but instead of actually doing very difficult chromatography and trying to isolate these molecules physically, we can actually do something smart, and I'm going to describe this now. So instead of having a single laser where the molecule diffuses and we build this one-dimensional histogram, we added some complexity. Instead of using one laser, we use two lasers, and we alternate the two lasers very quickly while the molecules diffuse in the confocal spot. So during this diffusion, we alternate between green excitation that excite the donor and red excitation that excite acceptor. We basically ask, does this molecule have an acceptor on it or not? And by asking that question, we can electronically later on remove all the events where there are no acceptor molecules. So in this case, we can define, instead of only Fred value as a one axis for this histogram, we are doing a two-dimensional histogram. We define another ratio, which we call S, or stoichiometry. And that really ratio tells us if acceptor molecules are there or not. And using this method, we are able to basically look at only the Fred relevant species and eliminate from the data donor-only cases and acceptor-only cases. I won't go into the details, the papers are cited and so on, but that really allows us to do very sophisticated spectroscopy and measure distances and distance changes. We call this method ALEX. ALEX stands for alternation of excitation, or alternated excitation. So back to our transcription machinery. I mentioned briefly this abortive initiation cycle. This was actually discovered where in the building I'm sitting right now at UCLA in the early 80s by a researcher named Jay Gwala. He did an experiment called footprinting experiment. In this experiment, you use chemicals to cross-link DNA to the protein. Then you digest all the exposed DNA. Then you release the bound DNA to the protein, and you analyze by gel electrophoresis the length of the protected DNA during this footprinting experiment. And what he found that during abortive initiation, you get a very sharp end that is protected on one side and varying lengths on the other side, which was a puzzle. During elongation, the enzyme has the same footprint, doesn't matter where he is on the DNA of the genome. Okay? But in initiation, the footprint is changing. And that means that the enzyme somehow is stuck to the promoter at some end and not on the other end. That was a big puzzle. Think about a Beetle car, an old Beetle car, not the new ones that were developed, after the Second World War. Those were air-cooled engines. You would heat up very quickly. Think about such a Beetle, Chipushit, going up a hill, and the engine shuts off and it slides back. And it tries again, and it slides back, and it slides back. And only by chance, one of those attempts, it manages to go over the top of the hill, and then it goes into elongation. So that's abortive initiation. And the reason why the system is built in this fashion, it's believed that this is giving more time for regulation, for feedback loops. So that you want to be absolutely sure that you need to make now a particular messenger RNA, and basically this abortive initiation allows you to have, if you want, a bullet in the chamber, and if you need, you can go, but you're spending a lot of time in that situation until you get the instruction to go. So you basically make those short abortive RNAs, which are useless, are being degraded, but you're basically in this situation. And after J. Gralla experiments, people have tried to figure out what is the reason for this and how the abortive initiation works. And we set up together with Richard Eberle to answer this question. At that time, when we approached this, we published our paper, and this is the paper you're going to get the exam on. That paper was published in Science in 2006. And that was about 25 years after J. Gralla's first report of abortive initiation. Over this period of 25 years, people came up with all kinds of suggestions for mechanism for abortive initiation, but nobody really solved that problem. So what were the three models explaining abortive initiation? The first model is called RNA inchworming. Think about the enzyme as a worm. One part of the enzyme is fixed to the promoter, and the other part is basically kind of flexing forward, starting to make the RNA, lose the RNA, and snaps back. So until the enzyme goes into elongation, the back part of the enzyme is fixed on the DNA, the leading part of the enzyme is moving forward and backward. This is called inchworming. The second mechanism that was proposed was DNA scrunching. If instead of the enzyme doing basically this conformations on the protein, the conformation happened not in the protein, but in the DNA. I mentioned before the transcription bubble. This model suggests that the downstream DNA is being basically moved into the enzyme. The DNA is kind of folding on itself, builds some stress, the RNA, short RNA is lost, and then the DNA snaps back. This is called inchworming. And the third model says it's not conformation in the protein, it's not conformation in the DNA, it's actually just the difference between single molecule and ensemble measurement. The enzyme sits most of the time in the back position, made very short, very brief movement forward, lose the DNA, the transcript, and slides back and sits most of the time in the back position. So when you do those ensemble measurements of footprinting, you see basically a sharp edge on the back side and smeared edge on the front side. So those were the three models that were there for 25 years. And really, nobody could use a regular method at that time to try to answer which of these models is the correct model. So what I'm going to show you next is how we use single molecule threat to answer which of these three models is correct. And the exam you're going to get today is basically based on this. Okay. So what we did, we basically labeled the DNA, we labeled the protein, we labeled the protein in the leading edge, we labeled the protein in the trailing edge, and also the DNA upstream or downstream. And we basically looked for conformational changes and to decipher which of those different models is correct. So again, let me show you again. This is going to test the motion of the leading edge of the protein compared to downstream DNA. This is going to test motion of the backside of the enzyme compared to upstream DNA. And this labeling is going to test changes in the distance on the DNA itself. So here comes the first experiment, which is a control experiment. We are doing what we call the leading edge threat. Leading edge of the enzyme is labeled as donor, downstream DNA is labeled as acceptor. All three models predict that during this abortive initiation, the distance gets shorter. So here is the crystal structure, the position of the dyes. Here is basically the open complex. You get a cloud. This is a cloud of the distance. Basically, this is a collapse of this into one-dimensional histogram. This is the Fred peak. And you add now nucleotides, and you can, instead of adding 4 nucleotides, the first 11 positions of the promoter are made out of only 3 out of the 4 nucleotides. So we can starve the enzyme from escaping into elongation. If we add only a mixture of 2 nucleotides or 3 nucleotides, we can march the enzyme to different positions. So you can see that once we give it nucleotides, we can go up to position 7 on the promoter. This peak now is shifted to the right. And as predicted, all 3 models predict that there is a motion of the leading edge compared to downstream DNA. Indeed, we measured this. So this is basically serves as a control experiment to show that the assay is actually working. Now, only 1 model out of the 3 predicts that there is a motion on the backside. This is the transient excursion model, where the enzyme makes a brief excursion forward and then comes back. And here is the measurement. So we start with this high Fred peak. We add nucleotides and absolutely no change in that position. So we can already eliminate 1 out of the 3 models. This is definitely happening. And we are left with either scrunching or inchworming. So to test this, we put donor and acceptor on the DNA itself. And if there is scrunching, that distance needs to get shorter. So here is the measurement. Already here, because we have the DNA labeled, we have some free double labeled DNA. This is this peak. There is always that contamination. But we have protein bound to DNA. Now there is a transcription bubble, so that distance is shorter. This is this second peak here. And if we add nucleotides, that cloud here is shifting to the right, and we very clearly see that shortening. So basically, by doing all those different type of experiments and using the unique property of single molecule Fred to look at conformational changes, we did many different positions, and very clearly, we demonstrated that this is the mechanism for abortive initiation. Since our work in 2006, there were many cryo-EM structures and many other works that really confirmed this. So now it's very well accepted that scrunching is a mechanism for abortive initiation. I'll very briefly go through another work of ours. Will not dwell on this too much. This has to do, once you get to position 7, there is a block. This sigma factor 70 that I mentioned, in particular region 4 in sigma 70, blocks the exit tunnel for messenger RNA. Here we start to make this messenger RNA, gets to position 7. You see the scrunching, and now the RNA cannot grow because the tunnel from which it's supposed to get out is blocked by sigma 70. Sigma 70 has to release some contacts with the core enzyme to open up the tunnel. And this is really the mechanism for abortive initiation, because if the tunnel opened, the transcript is lost, the enzyme slides back and try again and try again. So we wanted to look at this potential barrier. And the way we did this is published in this paper. This is much later. This is 2016. And here we did a kinetic experiment. So basically, if we start the enzyme at position 1, we get very fast transcription. But if we start the enzyme, I told you we can starve the system, march it out to position 4 or position 6 or 7, and then resume transcription by providing the full set of nucleotides. We actually get much slower kinetic. And that was really very puzzling. How come if you start earlier, it's fast, and if you start later downstream, you are slower? Apparently, there is a protein called GRIA that if there are some pausing and backtracking, RNA actually is going back to the point where nucleotides are added to the enzyme. And a protein acting as a scissor has to release and cut some of that RNA in order to build, again, a registry such that the enzyme can continue. If you use that enzyme, instead of getting this slow response, we now get back the fast response that we had if we started at position 1. So we basically proved in this work, again, using single molecule FRET, that there is a pausing in initiation, a pausing that was not known before. So we could revise the model for initiation. Before our work, there was this feedback loop for abortive initiation, and we have shown that there is an additional slow step, which is basically pausing and backtracking. I just want to finish today's meeting telling you that the single molecule FRET story is not over. It's nowadays a very, very popular method by many researchers. Here, for example, you see a paper published by a bunch of us in the community that was published in eLife last year. First, let's start with this paper. This is a review paper that my group wrote in Science in 2018. This is an eLife paper that was published last year. There are many fantastic developments in making single molecule FRET even better. This is a paper published in Molecular Cell last year by the group of Takjip. Takjip was my student, and now he's a very successful professor in Johns Hopkins. This is taken from Web of Science, just showing the adoption of single molecule FRET. More than 4,000 publications doing single molecule FRET experiments. More than 70,000 papers citing single molecule work, and 123,000 cases of citing single molecule FRET. There is a very bright future for this method. People have developed both open science instruction how to build, and until about 10 years ago, the method was really in the hands of experts who knew how to build those microscopes, write the software and so on for signal analysis. But this is now being democratized. This is open source 3D printing approach to how build a microscope like this. There are commercial systems by Peacock One, ONI, and EI. All kinds of new experiments that can be done that have not been done in the past. Instead of having a single spot, you can now do multi-spot analysis and much higher throughput using special spot array detectors. You can do single molecule FRET in individual bacteria in live cells. You can use light sheet microscopy to look at mammalian cells. You can do non-equilibrium reactions where you use microfluidics to mix, basically stop flow experiments for single molecules. Here you can see work where you use not 2 dyes, but 3 dyes, 3-color single molecule FRET, where you can get basically 3 distances simultaneously. The group of W.E. Morner developed something called the Abel trap. You can actually have molecules in solution, but you have a feedback loop to keep them in the center, so they diffuse, but the stage is moving to keep them always in the center, and you can do very long trajectory of experiments of molecules that are not immobilized on surface and are not perturbed. You can do fluorescence lifetime measurement together with single molecule FRET using pulse lasers and time-correlated single photon counting to look at very fast conformational changes. This is work from the group of Ben Schuller. And Eitan Lerner, who was my postdoc and now professor at Hebrew U, has developed really interesting signal analysis methods to look at dynamics faster than basically the diffusion time in the confocal spot. So there is a lot of new additions to the basic concept, and this becomes a major tool in looking into conformational dynamics of macromolecules. There is a whole community. If anybody is interested, you can sign up and be part of that community with newsletters, lots of activities, and so on. So this is a story I wanted to tell you today. What I'm going to do right now I'm going to post the paper on this week part of Moodle. I'm using Moodle, by the way, and the exam. You have until Sunday midnight time to answer. Please upload your PDFs for this assignment. Any questions for today's lecture? Any questions about the assignments?
Sunday midnight is the end of Sunday?
Yes.
All right.
Anything else? So I ask again, please pick up the topic, check with me, and put your date down on this Google Sheet. All right. Thank you, everybody. Good night. Bye-bye.
Thank you.